official BrainHack Ignite speaker for 2018. This is Emma. She's a Emma, who's a longtime friend from McGill and amazing scientist who is leaving Toronto for London in a few months. And she has some pretty actually amazing work with baby brains to tell us about. So I will let her take over.
Okay, great. Thank you, Erin. Thank you so much for inviting me to present today. So I'm going to be focusing my talk on different methods using data mining to the study of the developing brain. So I'm currently a senior research associate at the Hospital for Sick Children, and I joined to pursue my postdoctoral research studying brain behavior relationships in children with autism and children who are born preterm, who are very high risk for neurodevelopmental disorders. I also had the opportunity to work on some studies with children who have attention deficit hyperactivity disorder. So during the course of my studies, particularly with children with autism and those who are born preterm, I noticed that many of them had been critically ill at the time of birth. And this got me very interested in studying early adversity. So brain injury, stress, pain, and how this can impact the developing brain. And I'm now an investigator in the neonatal neuroimaging program and studying very preterm born babies. So these babies are born after about 6 months of pregnancy, so being born 2 to 3 months early. And the hallmark feature of preterm born babies is the very wide variation in terms of the developmental trajectories. So where some of these children can go on to develop common childhood psychiatric disorders, and while others can go on to really outperform term-born peers. And my research is focused now on looking at the different factors that can go on to predict alterations in the development of brain trajectories or predicting brain adaptation that underlies risk for neurodevelopmental disorders or promotes resilience. And very preterm born birth is quite common. It's about 1 to 2% of all births. So this is about 4,000 births in Canada each year. Very preterm born babies, they are spending the first few weeks of life in the stressful and hectic environment of the neonatal intensive care unit. So they're in an environment where the lights are on 24 hours a day. There's many invasive procedures. Many of these babies are very sick. They require surgery. And in turn, our group has shown that many of these environmental factors are associated with disruptions in early brain maturation. And this is also associated with adverse outcomes in terms of how they perform later on at school. So in terms of their cognitive functioning, sensory processing, behavioral disturbances. What's more is that preterm birth is associated with an increased risk for autism. So autism is a fairly common neurodevelopmental disorder with an early onset. So it's prevalent in about 1.5% of the general child population. However, for preterms, they carry a 10 to 20% risk of developing autism. Autism is characterized by alterations in social communication and also a number of restricted interests and repetitive patterns of behavior. And in terms of what's going on in the brain, children with autism, they often exhibit a pattern of early overgrowth followed by arrested development and disrupted structural connectivity. Preterm birth is also associated with an increased risk of attention deficit hyperactivity disorder. So ADHD is prevalent about 5% of the general child population, and preterm born babies, they have a 40 to 60% increased incidence of ADHD. So both autism and ADHD, these are disorders of early onset and associated with variable brain trajectories. And so how do we kind of go about predicting if a little infant baby, how are they going to develop and who is going to be at risk for these neurodevelopmental disorders? So the way in which we do this is we recruit babies while they're still in the hospital. So we recruit them and their families within the first few days or first few weeks of life. And we scan them with MRI. And we perform a scan around the time of birth and again at term equivalent age, so around the time they were intended to be born. And by having these two scans, this allows us to look at the growth of the brain. It allows us to look at the different factors like surgeries, medication, brain injury, pain, stress, how it can impact the different variations of growth. And then we follow these children till the second and third year of life and into school age with varying types of developmental assessments, so cognitive, language, motor functioning. And we use the information from the early scans as a way to predict outcome in terms of who is going to do well and then who may be at an increased risk for neurodevelopmental disorder. And so, as we all know, the brain is undergoing an exponential period of development from the kind of preterm period, around 25 weeks of gestation, through to term age and onto childhood. And for babies who are born preterm, they can be exposed to brain injury, stressors from intensive care and also clinical care, and this is associated with a disruption in brain development and also associated with poor outcome later on at school age. Yet at the same time, there's also preterm born neonates who end up performing really well. They can end up performing much better than term born peers. And so our research is focused on using different analytic methods as a way to predict outcome in these babies. And one method that I'm going to talk about today is probabilistic mapping. The probabilistic mapping is a way of collating data from different studies, different imaging studies. You can collate data from structural studies, from functional studies, and this allows you to look at the spatial coherence of a particular cognitive function or alterations in brain morphometry to improve localization about a particular brain region or about functional activation associated with a particular task. And so for my talk today, I'm going to be focusing on different types of probabilistic mapping and how we can use this information to the study of the developing brain. And so this is a revamped slide. I'm sure many of you have seen this image. For those of us working in neuroimaging for a long time, we always had this, we saw this slide from the late 80s. And so it's about the spatial and temporal resolution of different functional brain mapping methods. And so I'm going to be focusing on electrophysiology, which is one of the original brain mapping methods used by neurosurgeons like Wilder Penfield at the Montreal Neurological Institute. So, and that's, down here. And I'm also going to focus on probabilistic mapping of brain lesions and functional fMRI and one structural MRI study. So very preterm born babies. So as I mentioned, their brain is undergoing this exponential period of development during the period while they're in the neonatal intensive care unit. So this is an example of a scan. This is a T2 weighted image acquired of a baby around 29 weeks of gestation. And what you can notice, so these are, the eyes are here, so this is the front of the brain, back of the brain, is that there is, relatively, there's no gyri, it's relatively smooth. And then as we get to the time when the baby is intended to be born at 40 weeks, this is where we see the adult features of the brain start to come online, where you can see all the gyri and sulci. And so this is happening while the babies are undergoing intensive care, where they're being exposed to many different types of stressful, invasive procedures. And so their brains can be impacted by the indirect effects of environmental stressors, but they also can be impacted by the direct effects of brain injury. So brain injury is highly prevalent in these preterm born babies. So it's about 30% of babies that will have brain injury. And it's associated with poor outcome and even severe motor impairment like cerebral palsy. So this is the major risk factor for cerebral palsy is white matter injury, so damaging the white matter or fiber tracts. And this is what it looks like. So this is a T1 weighted image acquired in a neonate. It's a sagittal view. And the white matter injury are these bright spots here impacting the white matter. So, of course, the brain isn't really myelinated very much at this point. It's really kind of only myelinated up until the up until like the midbrain diencephalon area. But we still see this white matter injury. And so for right now, in terms of using MRI as a way to predict outcomes, so it's not routinely done in these babies. And for physicians who treat the babies, if they don't know what to do with this information. So because there is babies who will have this white matter injury, and there'll be no association with cerebral palsy. The children will end up doing fine. So in terms of the clinical utility of using imaging as a way to predict outcome, it had never been up to this point really that evident. And so when we started to kind of tackle this problem of how can we predict outcome using this information about injury, we looked first by doing a diffusion tensor imaging study with a group of very preterm-born babies. And so we scanned them, and the families returned at when the infants were 2 years of age, and they were assessed on standardized tests for cognitive, language, and motor function. And so we performed track-based spatial statistics, and this allows you to look at alterations in white matter microstructure on a voxel-wise level, and that's what's shown in orange here for the maps. And so we then extracted some of the DTI metrics, so fractional anisotropy values here, and we then plotted it in relation to the outcome scores. And so the question we were asking is, if you perform diffusion tensor imaging on a baby, on a preterm-born baby, can we use this information to predict who is going to end up doing well later on? So can we use FA as a metric of outcome prediction? And the answer is yes. So it was strongly correlated with cognitive performance later on in life. And so for the babies who are performing well on this standardized test, so they're performing way above the mean of 100, they have the highest fractional anisotropy values. And the babies who are performing the worst, so they're performing more than 2 standard deviations below the mean, so which is a clinically significant sign of deficits, they have the lowest FA. So we said, oh, okay, well, these must be the babies with brain injury, right? So if you have white matter injury, you damage your white matter microstructure will be affected. You're going to probably have poor outcome. But the answer is no, so you don't. So we took the babies out of the analysis, and there was no real association with brain injury and the fractional anisotropy data. So then we said, well, is white matter injury really predictive of outcome? So we were left with the question of how do we take this information from the early MRI scans, and how do we use it to predict how these children are going to do later? So to answer this question, we turned to probabilistic mapping. So I returned to the electrophysiology literature. This is one of the original brain mapping methods. And during my master's degree, I worked in atlas development, in functional atlas development. So I'm sure many of you are very familiar with the use of atlases now. So people are using the AAL atlas applied to cortical thickness data. You can use it for for region of interest analysis, for functional data. But you can also use information from atlases to do neurosurgical planning. And so this is an atlas that was developed for neurosurgical planning. So this is applied to the basal ganglia region. So this is, I think it's probably easiest to see in an axial view. So this is the thalamus. These are the internal capsules in blue. The different colors represent the different thalamic nuclei. And so these atlases are used for the placement of deep brain stimulators. So deep brain stimulation is used quite commonly now for the treatment of movement disorders and Parkinson's disease. It's also used as a wide variety of applications. It's used for chronic pain. It's used for obsessive compulsive disorder. And essentially, these stimulators are inserted into the brain depending on the different disorders. So for Parkinson's disease and movement disorders, it is inserted into the basal ganglia region. And then for chronic pain, it's the motor cortex. So but the actual placement of these stimulators is done entirely through image guidance. So the placement of the stimulators is done by just drilling a very small borehole into the skull. And so to do that, they use they first put a stereotactic head frame on the patient, so the patient's head will go here, and this is where the tools, the surgical tools, will go. But by building a head frame, this provides a coordinate space. So then you then have X, Y, Z coordinates in the brain. So this is how neurosurgeons are able to do this, to only have a tiny bore hole drilled into the brain and place a very tiny electrode into the brain. So they put a stereotactic head frame on, they scan them with MRI, and then they co-register the MRI image with the frame space, and then you can have your 3D MRI, and then you can essentially look inside the patient's brain. And you can apply atlases. So you can register an atlas to the brain, and then you can use surgical planning tools, which is shown here. So this is a 3D representation of an atlas on an axial image of in a patient brain. And so this, so down here, these are the globus pallidi. This is the caudate nucleus. And you can see that the, in green, this is the this is the DBS stimulator being implanted into the basal ganglia region. And so in addition to having these atlases, what neurosurgeons will do is they will perform electrophysiological mapping as a way to localize where they are in the brain. So they'll insert a stimulator, and then they do electrophysiology. And in surgery for Parkinson's disease, they will can insert a stimulator into the thalamus, and they will stimulate the internal capsule. So this is the motor pathway shown here. And you can see here that this is the representation of the motor homunculus. And the internal capsule is very much like what we have in the cortex, and that is also organized where you have the body part representations. And so neurosurgeons will use this information about stimulating the internal capsule, and they know the organization, and in turn they're able to localize where they are. And so this is how we would build functional atlases from the data obtained in awake brain patients undergoing brain surgery. So this is a 3D MRI rendered. You can see the ventricles in purple here, and the thalamus is blue. And a virtual electrode is being inserted into the MRI. And in turn, the neurosurgeon is going to be stimulating the internal capsule, which is just lateral to the thalamus. And how that looks is for each microelectrode point, there will be a motor response in the patient. So it's a very small motor movement. And so the surgeon will give stimulation, and then they'll actually have the a response that will occur in a somatotopic organization. And so you can record this information with in the little homunculus here. So if you have a stimulation point in the internal capsule, there'll be a movement in the leg, and then you can then tag that as a leg response. And by doing this, you can in turn map out the different body part representations. So this is what it would show. This is what an image of the internal capsule would look like with the different body part representations. And this is an average. So this is a 3D MRI with an atlas overlaid. And we can see that when you look at the average body part representations, this is in the posterior limb of the internal capsule. You can see there's this nice somatotopic organization in the internal capsule area. And so looking at the kind of the average localization is important. You know, this can tell us information about anatomy, but it's not very useful in terms of looking at the kind of the full spatial extent of the data. So what we then later did is we created probabilistic maps of the three-dimensional electrophysiology data. So we pooled all of the data together from all of the subjects. We then registered it to a common template, so all of the data were in a common MRI space, and then we developed probabilistic maps. And so this probabilistic map is telling us that there's a value from 0 to 0.005, indicating the likelihood of obtaining a face motor response when stimulating any part of the brain. So of course it's localized to the internal capsule. So you can do that as well for the different body part representations. And so when you look at the mean points, you can see that there's a reliable organization from face to leg. But then when you overlap them using the probability maps, you can see there's quite a bit of overlap between the two representations. So all being to say that this is, you can use probabilistic mapping applied to your data, to a large data set, and this in turn can allow you to have insight into the spatial location in a common 3D space. So then, returning to the question that I started with of how can we predict outcome in a very preterm-born baby, we can use the same method used in electrophysiology applied to the lesions. And what we did is we, just how I had created those functional databases, we labeled each and every single white matter injury point in the T1 weighted MRIs. Well, I didn't do this. We actually had a medical student do it painstakingly for a year. And so she had labeled all of the white matter injury points in the native space of each baby brain. We then created a template, and then we then combined all of the maps together, and this allowed us to assess the probability of the location of the injury where there was occurring. So is there a pattern? And the answer is yes, there is a pattern of injury. So this is a cohort of 250 very preterm born babies who were scanned within the first few weeks of life, and they then returned in the second year of life for neurodevelopmental follow-up. And what this image is showing here is the pooled map from about 60 babies, in which it's showing you with the warmer colors is where there was overlap in terms of the lesion location. So in about 20% of these babies, they had the same lesion in exactly the same location in the brain. And this was in, so if you look here, this is the central sulcus. And where we see the peak, this is in the white matter fiber pathways underlying the sensory motor cortices. So indicating that white matter injury in babies is not random, it's occurring in a repetitive pattern. And then, as a next step, we said, how can we use this to predict outcome in terms of, is a lesion located in one of these areas of the brain associated with an adverse outcome later on? Or is it just the total size of the injury? If you have kind of massive brain injury, is that predicting outcome? And so we went one step further, and we had the outcome data from those standardized tests that I presented previously in the diffuse intentional imaging study. And this is an odds ratio map. Is a brighter color indicates poor outcome. So, and I think this is really nicely illustrated for the cognitive maps. So there's actually only a couple of voxels in the brain where it was associated with poor outcome. So indicating that if a baby has even a very small lesion to this particular area of the white matter, they have an increased risk of poor cognitive outcome later. But there is no association with total lesion size. So even if they had a very large lesion in the back of the brain, in the posterior part of the brain, it was not predictive of outcome, which is really counterintuitive. Would you think more injury, the baby is going to do worse? But the answer is no. Even if you have a tiny, small lesion as a baby to this area, that is what is associated with poor outcome. And so the next question that we wanted to ask following up using this probabilistic mapping of lesions is, how is it predicting impacting the developing brain? And so this is a coronal image of the developing brain. And this is the thalamus here, globus pallidus putamen, and this is the cortex. And in blue, this is representative of the white matter fiber pathways. And in the preterm-born babies, their brains aren't myelinated yet, so they have the precursor cells to the oligodendrocytes, which wrap around the axons to form myelin. They have these precursor cells, and they're very, very sensitive to oxidative stress. And this, by damaging these cells, this is what causes white matter injury. That's what we see in the T1s. So we wanted to ask in a new cohort of babies, in a prospective cohort, how does white matter injury impact functional and structural connectivity? So we had the babies, we scanned a cohort of nearly 50 babies with of course, structural MRI, and also in functional MRI resting state. So resting state is the same in the baby brain as it is in the adult brain, just the networks are less developed. And we also did diffuser tensor imaging. So the first thing we did is we created the probabilistic maps, and we found there's a very similar pattern to what we had reported in that other cohort. And then we looked at the resting state data, and we found that the babies without any type of brain injury, they have kind of robust bilateral networks. And then in the infants with white matter injury, we see there's more unilateral activation opposed to what we see more bilaterally in the non-injury group. We then looked at the strength of the functional connectivity between the thalamus and the cortex. And that's what's plotted here on the y-axis, is the strength of the functional connectivity between the thalamus and the parietal cortex. And then we looked at that in relation to the total volume of injury in the different white matter tracts. And we found that if you have decreased functional connectivity between your thalamus and parietal cortex, you have increased white matter injury. And we also extracted the fractional anisotropy values and found there was also a significant negative association with a greater volume of injury. And so, using this probabilistic method, we were able to understand, by probabilistic mapping, we were able to better understand how it can impact the developing brain. And so the next step for this cohort, they've since turned 2 years old, and so we're bringing them back still, and the next step would be looking to see how we can use this information to predict outcome. And so that's for the using probabilistic mapping for the infant brain. So for the next part of my talk, I wanted to focus on ways of using probabilistic mapping to answer more complex questions in children with autism. So children with autism spectrum disorder, as I mentioned previously, they have social communication and interaction difficulties, and they also have restricted and repetitive patterns of behavior. So an example here is a child who is showing lack of creative and imaginative play, and instead is lining up his toys according to size and color. In addition, children with autism also have a number of sensory disturbances. There was about 20 or 30 years ago, autism was considered a very rare disorder, and now we know that's not true. It's actually very common. There's a report that came out last week from the CDC to say that autism is now prevalent in 1 in 59 children, so it's a very common disorder. But there was a lot of interest in doing post-mortem studies 20 years ago or so. And so many of these studies, they contained the post-mortem samples of, just a handful of brains. It was, 3 to 5 brains. And so there was, a great deal of interest in, studying these post-mortem brains. And so even though it was only based on, 2 or 3 individuals, this became kind of very common in the literature to refer back to these very few studies. And so these studies reported that there was enlarged frontal and temporal lobes, and there was also alterations noted in the anterior cingulate cortex. Many people began focusing on this enlarged brain hypothesis in autism. And there is some credence to this. So this is a study done with over almost 700 children. And this is head circumference measurements. So as children have their regular appointments with pediatricians, they'll have their head circumference taken, their birth weight and length. And so they had access to this enormous data set where they had these head circumference measurements. And they then looked to see who then was diagnosed with autism at 2 to 3 years of age, and found that there was incidence of having increased head circumference. This is associated with autism. And so for people who are doing neuroimaging findings, who are doing neuroimaging, you could think of this as very kind of crude measurement, but it's very easy to do. You don't need to spend $1 million or $2 million on installing an MRI, so it could be a kind of an accurate measure for an accurate prediction measure. So there was certainly people certainly followed this up though in neuroimaging studies. So this was published not that long ago. And this is almost 100 children, and this was a longitudinal study in which they acquired 2 time points. And they looked kind of globally at the total cerebral volume, gray matter, white matter, and found that the children with autism had greater volume. So this was kind of in line with the enlarged brain hypothesis. So imaging studies and also postmortem studies were kind of pooled together. And this hypothesis about this early overgrowth occurring early on in infancy, followed by arrested growth and then possible decline later on in adulthood, this is a theory that's often referred to in the literature. And so to answer this question about brain development and autism, so I did my postdoctoral work in autism, and people would often ask me about this overgrowth hypothesis. And I thought, you know what, I don't think we really know, looking at these postmortem studies. And so what we did was a meta-analysis of structural imaging findings in autism. And so we did it on voxel-based morphometry studies, so which is tells you about voxel-wise differences in density. And so meta-analyses, it allows you to look at contiguous activation patterns or changes in morphology associated with a particular clinical group or a particular task. And it allows you to overcome, many of the limitations of single studies, so having few number of subjects. Also, in the pediatric literature, it's very difficult to scan children because they often don't stay still, so you have to throw out a third of your data. So by collating data from many different studies, this allows you to kind of look across different study population groups. You can do it for structural imaging, and you can do it for functional imaging. As long as you have 3D coordinate points, then you can pool them all together, just like how I had presented for the electrophysiology data and for the lesion data. You can pool it all together and look across as many studies as you would like to be able to answer this question. And that is what we did, so we used the ginger ale method for this analysis of the structural changes in the brain in children and adults with autism. And so they used to call it the activation likelihood estimate method, and then they revamped ginger ale to do structural studies, so then they call it the anatomical likelihood estimate method. And so this is a freely available software, and you can download it, and you can do a meta-analysis quite easily. You can just pool together your studies as long as they have data in a coordinate space. So you have to painstakingly review your studies, find out what coordinate space they were done in, MNI or Talairach. Then you have to just pick one space and convert your coordinates into a single space. And then using the software, they will develop probabilistic maps and is testing the peaks against a random floor of noise. And you get a probability value in the brain showing a likelihood of having activation in a particular region associated with a functional task. Or in our case, it was looking at alterations in density. And so we did this meta-analysis of 19 voxel-based morphometry studies. And we looked at both children and adults. And we found that there was both increases and decreases in the child and adult brain looking across these different studies. So what would have been predicted is that in the adults is that you would only see decreases in volume, and that in children you would potentially only see increases. And so in actuality, when we looked across all these studies and pulled them together, we found that there was both increases and decreases reported. So we looked at the gray matter and white matter changes, and the data in orange are the children and the data in blue are the adults. And we found that there was evidence for increases in some areas in the children, but as also decreases, and the same for the adults. So it certainly didn't necessarily fit with this original hypothesis, certainly about the decline. And now this is before there was the ABIDE data set. So there's these large data sets, collected structural scans collected in hundreds of subjects. And now there's ABIDE 2. It's a grassroots project, so it's people donating their data, but they're all acquired at 3 Tesla. And so you can access these large data sets. And I'm sure people are being able to answer these questions looking at 700 scans. So ABIDE 2 is new. Is there ABIDE 3? Yeah, so it's a 2000. So I'm sure there's people who are working on this hypothesis. And so, but by using probabilistic mapping, this is a way of collating findings across many different studies, to answer this complex question. And so about structural changes. And so in addition to using probabilistic mapping to answer complex questions about structural alterations in children with autism, we can also use it to answer questions about alterations in the core morphology, the core features of children with autism. So as of late, there's a relatively new hypothesis, which is rethinking autism and ADHD and many of these neurodevelopmental disorders in children. And the thinking is that perhaps it's kind of all of a similar disorder, and that many of the behaviors that we see in these children may be a reflection of alterations in the ability to process salience. And so, for example, children with autism, the difficulties with social interaction and communication, this may be difficulties with recognizing salience of emotional faces, restricted and repetitive patterns of behavior. This could be reflected in focusing on internally generated stimuli. And so many groups have looked at this and have started examining the possibility of there being alterations in the salience network, and this may be common to many different disorders, childhood psychiatric disorders. And so for the salience network, it's thought to be comprised of two core brain regions, including the anterior insula and the cingulate. And so when we have a salient stimulus coming in to the environment, this in turn turns your salience network on. So this could be a loud noise, or it could be touching something very hot. It turns off your default mode network, and then it turns on your central executive network to say that you have to pay attention to this salient stimulus. And so it's thought that potentially in children with autism, there could be disruptions in the salience network, where if they have difficulty with processing information about in the environment that's salient, and they miss this switch of turning on the central executive network. And so we did, our group did a number of studies looking at altered salience processing in children and adults with autism, doing a number of different studies, looking functional studies, structural studies, and also doing psychophysical studies, looking at sensory processing. And while I won't go into the details of those, I'll just focus on two main studies that were done in the same individuals, looking at altered emotional salience processing in children and adults with autism. And so the first study that we did was a functional MRI task. This is a classic executive functioning task where we're looking at the ability for individuals to inhibit a response. So it's a no-go task. So we have, the individuals with autism are lying in the scanner, and they are getting presented these images of emotional faces. And they are being presented on the screen for only half a second. And they are trained to press a response when they get a go stimulus, so that could be a happy face. And then they have to not go when they see the no-go stimulus, which is a sad face. It's a very rapid task. And we wanted to use emotional faces, as this is kind of a natural stimuli that's used in the environment. And so individuals with autism, they can often have difficulties with inhibiting socially inappropriate responses. So we wanted in this task to look at the brain regions involved in processing emotional faces. And so this is a fMRI study, and then we looked at the activation for the no-go versus the go trials. And in the typically developing population, we see that they activate the salience network that I presented previously, anterior insula, cingulate. And then the individuals with autism, so they did the task, so just fine. There's no difference in performance, that they activated very different areas of the brain. And notably, they did not activate the salience network. We then performed a cortical thickness study on the scans and surface area. And we extracted the surface area from the cingulate and from the insula. And we found that the individuals with autism, which is shown on the on your right here, that they had greater surface area in the insula. And this was associated with social communication impairments. So the individuals with autism are in purple, and the typically developing group is in black. And so the individuals with autism who had the greater surface area, this is in the younger children. And so we see that there's this disruption in the development of this brain region, and this both the insula and the cingulate, the key structures involved in salience network, were associated with disruptions in social communication impairments. So impaired emotional salience. And so this is what kind of single studies can tell us about something very complex. But then you can also do this using meta-analysis. You can also view single studies, and you can collate this information across studies to answer very complex questions. And so this was done in a meta-analysis in which they looked at in typically developing and in children with autism, and they looked at all different fMRI studies, and they looked to see the likelihood of activation associated with social and non-social stimuli. And so, for example, here, so NC, this is in the controls. They found that there was increased likelihood of activation in the anterior insula for social stimuli, so key region involved in salience network, and the same in the cingulate. But in the ASD population, they don't see likelihood of activation in the cingulate or insula, indicating in this larger study looking across individuals with autism that there may be credence to this impaired saliency hypothesis, and this may underlie core deficits in autism. And so with that, I'll just summarize, just saying that through probabilistic mapping, this can provide a quantitative approach. It allows you to have improved localization, and you can perform predictive analysis using probabilistic mapping. And this it can allow you to study very complex questions applied to childhood psychiatric disorders and to complex cognitive processes. And you can also use this information for future hypothesis-driven research. And with that, I'd like to thank my collaborators, and I'd like to thank you for your interest, and I would be happy to take questions at this time.